I'm going to tell you today about decision making and specifically assembling information to guide decisions, when it works and how it can go wrong. And I've tried to set up the talk today so that it will be accessible to a pretty broad audience, to folks with quite diverse intellectual backgrounds. So do feel free to, in the Q&A, press me to dig deeper into any of the issues if you'd like to know more. So I think first we should maybe start with a definition. So what is a decision? Well, for today, our operational definition will be this. A decision is a commitment to one action at the expense of other actions, or sometimes of inaction. So an example here is this cyclist who's cycling along and is facing a fork in the road and must make a decision whether to go to the left or to the right. And obviously, as you can see from the image, the two different decisions have rather different consequences. And this observer will be taking in the sensory information and some other stuff and making a decision about what to do. So what sorts of decisions are interesting to study? Well, so here's the thing. So as you might have garnered from the slide before, decisions are naturally going to vary along a continuum of complexity, right? So at one end, we have really simple sensory motor reflexes, like maybe the knee-jerk reflex that you do with the doctor, that's so simple that we probably wouldn't really call it a decision at all. And then at the other end of the spectrum, we have decisions that are totally complex, like, for example, deciding whether or not to attend a lecture at the trailing end of the Omicron wave, or something like that. And so in order to think about which decisions to study, we want to kind of find something that's in the middle. And so for me, I really want to understand the neural mechanisms that support decision-making. So the key thing then is to find a decision that's simple enough to be able to be studied in a laboratory context, but complex enough to be interesting and to really tap into intelligent behavior and telling us how intelligent organisms are able to take in information and do something with it that's interesting and clever. So for me, one feature of decisions, I guess you'd say, that makes them interesting is decisions that assemble information, that is to put together or integrate multiple sources of information. And there's lots of ways you might do that. So some decisions assemble information over multiple sensory modalities. So in this example here, the mouse can get auditory information from the cat's paw on the floor, and also visual information by seeing what direction the cat is moving. And the animal might integrate those two sources of information to make an estimate about what direction that cat is going to move. And that's obviously a pretty important decision because it could be life or death for the mouse that has to plan its own escape route. Other sorts of decisions might assemble information over time. So this is really the same idea. Instead of putting pieces of information that are coming from different sensory modalities, you might put together pieces of information that arrive at one or maybe more modality over time. So here's an example of a non-human primate that's making a decision about the direction of dots that are randomly moving on a screen. And because the dots are moving stochastically, it would be a smart strategy for the animal to integrate over time and maybe over space as well to get a general idea of what was the overall direction of motion in the stimulus. So these two examples that I just gave are both examples of where decisions are made about purely perceptual stimuli, right? You're either putting together sensory information from, say, vision and audition, or sensory information that's arriving over time. But of course, in real life, decisions might not only involve sensory information. Instead, diverse sources of information might be combined. So if we return to our cyclist for a moment, she might take in the sensory information from the dog, right? Maybe the size of its teeth or how terrifying its bark sounds. But she might also have a lot of internal signals that she can combine that sensory information with, right? So she might have some prior knowledge about how dangerous that particular breed is, or she might be able to pull from memory a prior experience with that particular animal. And that would guide her decision, and she could combine that with the sensory information to make a really good decision about what to do. Okay, so why is this interesting? So why do we study decision making? Well, I think for me, there's really a couple of reasons. So first of all, there's relevance to a lot of clinical problems. One example is addiction, which to some extent is certainly a disease of decision making, right? Many times people want to stop taking the substance that they're addicted to, but they're not able to make that decision. Studying decision-making is also informative about cognitive processes that can be genetically altered, and a couple of examples of those are attention deficit hyperactivity disorder, ADHD, and also autism spectrum disorder, in both humans and animal models. And that finally, I think the study of decision-making can be an entry point to understanding intelligence and which components of intelligent behavior are shared across humans and animals, and also what neural mechanisms support that kind of intelligent behavior, maybe similarly or perhaps not in different creatures. But you'll notice that there's kind of an assumption here with these last two, is that they both focus on humans and animals together. And that's in a way sort of an assumption that suggests that we might reasonably learn about one creature by studying another creature. We might learn about humans by studying animals, for example. And I think this is a hypothesis that when I started out my own lab, I really wanted to test whether or not this was true. And to do that, my first goal was to study the same decisions in humans and rodents. And in a sense, what we were doing was not particularly new, right? After all, certain kinds of rodent studies that people have been doing for a long time have some relevance to humans, right? So for instance, if you have a rat in a maze, that's presumably informative about humans navigating in a real world environment, but the connection between the two is a little bit vague. And so we wanted to have something that was much closer. We really wanted to put the human and the animal subjects head to head, be able to compare them using the same tools, and really see whether the same decision-making mechanisms were at play in both species. So to do that, we decided we would start by really scouring the perceptual literature and looking for multi-sensory decisions. Remember those that put two different sensory modalities together to guide a choice. Find multi-sensory decisions in humans that we could realistically bring to a rodent model. And so we looked at a lot of different types of studies and interesting multisensory behaviors and kind of ported them into our lab and changed them a little bit. So we made a decision task where humans and animals judge the repetition rate of a stimulus. It's either a series of flashes or a series of clicks, or sometimes both together. And we could study these in both species because in humans, they could report their decision about the stimulus rate by a key press on a computer. And with the rodents, they could report their decision about the same stimulus by making a movement to either a left or a right choice port, indicating their choice by breaking a little infrared beam that was in the center of the port. And I should say that when we started off doing these experiments, mostly people didn't think this was the best idea that they'd ever heard of. And I think the reason is because multisensory decisions are quite tough. Part of the reason that this was studied in the human perceptual literature is because multisensory decisions are quite clever and skillful. Humans don't just sort of throw together multisensory information willy-nilly. They do it in a really strategic way, where they take into account how reliable each sensory modality is, and then use that knowledge of reliability to put the two signals together and ultimately guide action. And so I think people felt, well, that's a lot to ask of a rat. Is it really feasible to see these same kinds of optimal cue combination, multisensory combination in an animal like a rat? But in my lab, we wanted to find out. So we trained up rats on the same task that we were able to present to humans in our lab, and I'll show you a video of what that looks like now. So every time the rat goes into the center port here, it's presented with a series of clicks or flashes, or sometimes both together. And it's learned with lots of experience that high rate means go to the right and low rate means go to the left. And I'll show you what that looks like now. They get a liquid reward if they get it correct, and these are all correct trials. So it turned out, interestingly, that the rats were pretty willing to do this task, and they're willing to do around 1,000 trials a day, which is really nice because it gives us a lot of kind of data to play around with when we're trying to fit different models and compare the rats and the humans. So what I showed you there were almost all unisensory trials, so only the auditory stimulus or only the visual stimulus. But of course, we can put both of those together and sometimes make one more reliable than the other, allowing us to ask this question about whether humans and rats are both able to combine the information skillfully according to which source is more reliable. So we wanted to answer this question: can subjects flexibly change the weight of visual information? That is, how much they use the visual information to guide the choice. So the way that we look at the data is this. By fitting a number of models, we calculate the change in visual weight, that is how much weight each individual subject assigns to the visual stimulus. And in this particular configuration, anytime this has a higher value than 0, that means that the subject did the right thing, because this is for trials when the visual stimulus was reliable and the auditory stimulus was not. So anytime the value is above 0, that means the subject is doing the right thing. I'll show you the data for the humans first, and you can see that for all of the human subjects that we tested, except for one who's kind of an outlier, we could talk about him in the discussion, they almost always did the right thing. So they gave more weight to the visual stimulus, showing us that on a trial-to-trial fashion, they were able to figure out which stimulus was better and weight it accordingly when they put the pieces of information together to guide their decision. So now it was time to see what the rats did. And this is where the advantage of our approach really became clear. We didn't kind of have to extrapolate from rats in mazes to humans, walking in the streets. We really could just calculate exactly the same thing for the rats that we did for the humans, the change in visual weight based on a situation when the visual cue is more reliable. And we found that for the rats, just like for the humans, all of them changed visual weighting in the right direction, and both human and animal subjects were really close to the optimal prediction. So this to me was really interesting, and it suggested that decision-making computations can be shared by diverse brains. Now we know, of course, that human and rat brains are different in many, many ways, but I think this was, to me at the time, was very encouraging because it suggested that we might learn something about what I consider to be quite an intelligent behavior, that is the flexible weighting of evidence. We might be able to gain insight into that by studying the brains of non-humans. And that's a big win in the laboratory because we have a lot more tools to measure and also manipulate neural activity using animals. But before we started to measure and manipulate neural activity, we continued on with our behavioral studies for a while. And one thing I want to tell you about before I go on to the neural data is how do decisions go wrong? And I want to tell you kind of 3 common ways that decisions go wrong, that humans and rats end up making the wrong decision. And maybe this will help you moving forward in your own life to try to avoid mistakes or at least be forgiving if you make them. Okay, so the first thing that happens is that sometimes the evidence is just unreliable or conflicting. So I'm going to try to show you an example of that now. So your goal of the observer is to tell me by raising your hand whether you see 1 flash or whether you see 2 flashes. Okay? So hopefully you're all ready. There'll be 2 trials. Trial number 1 is about to start. Raise your hand if you saw 1 flash. Okay, very good. Okay, so a lot of folks felt that they saw 2 flashes. Hang on, let me just put that out. A lot of folks felt they saw 2 flashes, but oh, now it won't stop. Can you press pause on the movie in the back? Please. Okay, I'll go to a different slide. I'll go through that quickly. Okay, so even though many of you saw 2 flashes in the second presentation, in fact, the visual stimulus was exactly the same. In both cases, there was only a single flash, and what differed between the 2 trials was whether there was 1 beep or 2 beeps. And when there are 2 beeps, we have the illusory incorrect perception that there are also 2 flashes. So this is an instance where the sensory evidence is not only unreliable, but also conflicting because vision and audition are telling you different things. It's also unreliable because this is a task, judging 1 versus 2 flashes, that really relies on very, very precise timing. And in fact, your visual system is not really all that good at precise timing, partly because the retina is pretty slow to change the incoming visual information, photons, into an electrical signal in the brain. And so as a result, the visual system is not good at judging timing, but the auditory system is. So when you're faced with an unreliable visual signal and a reliable auditory one, you tend to go with the auditory one, which is exactly what happened here. So in this case, pretty much everybody made a mistake, but you can't really blame yourself, right? Because the sensory evidence was bad. Okay, so we'll try that again. Hopefully the movie won't play again. Can you please stop the movie? Okay. The other reason that sometimes subjects make mistakes is because they're using a strategy called Thompson sampling, in which they trade off safe options with potentially risky and rewarding ones. I tell you what, I'll tell you the decision-making errors on the next slide. Okay, so, and again, this leads subjects, it shows that the subjects have a trade-off between exploration and exploitation. And even though it leads subjects to make errors, even sometimes on very, very easy trials, which we would often call lapses, it actually represents an overall pretty good strategy, which helps subjects to explore potentially rewarding options that could be better moving forward. And the last reason that sometimes subjects make mistakes is that decision-making, even over the course of a short amount of time, like say an hour, which is about a typical length for a decision-making session, decisions are not stationary. So individual human and also animal subjects go through periods of time where they're quite engaged and make good decisions, and then other periods where they're using the sensory information in quite a different way. Sometimes the sensory information is ignored or it just has a weaker impact, and sometimes the decisions are biased, suggesting that the sensory information is being combined with some kind of an internal signal in a way that it really shouldn't be. And in fact, I wrote a paper about this recently with a colleague of mine, Jonathan Pillow, and Zoe Ashwood, who's a graduate student in his lab as part of the International Brain Laboratory, which is funded by the Simons Foundation as well. And so we discovered that this non-stationarity of decision-making, and interestingly, once again, we found that this tendency of subjects to go in and out of engaged and disengaged states was very, very similar in mice and also in humans. So this again suggests that even though mice, rats, and humans are very different animals, there are quite similar mechanisms that are at play in all three species. So having thought a lot about the behavior and why decisions sometimes go right and are clever and intelligent, and also why they sometimes go wrong, we were really interested in understanding the neural circuits that supported these kinds of decisions. And in my lab, when we first got going with this, we started off using a traditional approach where we were measuring neural activity, usually in one place or another at a time. And we learned a lot from this approach about multisensory decisions and I think decision-making and neural coding in general. But in a way, we weren't really satisfied. Because the problem was, when we're only recording from one or maybe two areas at the same time, we get a very limited picture of what's happening across the entire brain, even across the entire cortex. And for decision-making, which surely recruits diverse areas across the entire cortex, this is really problematic. Decision-making probably relies on the coordinated effort of multiple structures. So to only see one of them at a time is really very limiting. So we realized what we really wanted to do to understand decision-making was to be able to get a view of neural activity across the whole dorsal cortex at the same time. The problem was we were really not set up to do this at all. So our goal was to study decision-making activity very broadly, but what we were really set up to do was to measure activity in one place at a time. So in order to get going on this new goal, we had to retool the lab, and we started off making a checklist. So the first thing we had to do was to find a way to measure neural activity across the cortex. We had no way to do that. Second, we had to figure out if animals can still make decisions in this new setup. And I'll tell you in a moment why that was a challenge. Next, measure neural activity and behavior together in the new setup. And finally, analyze it to see if it makes sense. So this was our approach, and we'd hoped to tackle some of the questions relating to the behaviors that I described to you a moment ago, and we sort of have, but I'm going to give you a bit of a spoiler alert, which is that this approach told us something that we really did not expect at all about decisions in the brain. Okay, so point number one in the checklist is to find a way to measure neural activity across as many brain areas as we can. So we decided to use animals that have genetically encoded calcium indicators. And what that means is when their neurons fire, the neurons turn green, and they emit photons, and we can catch those photons with the camera that we have at the top of a wide field macroscope that we build in the lab. And we illuminate at two different wavelengths, one of them to activate the calcium indicator so that we can see this proxy for neural activity, and another one at a shorter wavelength to allow us to do a hemodynamic correction so that we can isolate the neural signals from the blood flow signals that occur at the same time. This is actually a rather easier setup to build than, for example, two-photon microscopy. And since we wrote a paper about this, it's become quite a popular approach. Many, many people were emailing us, asking us how we did it. So we ended up writing a protocols paper on the subject. So if you're interested, you can build one yourself, having read our paper. Okay, so I'm going to show you what the activity looks like now in a wide field imaging setup. And I'll show a number of these movies in the coming slides. The scale bar on the right shows you the change in fluorescence. That's kind of a proxy for how much the neurons are firing. And you can see that we can measure this quantity at every pixel in the brain. It has about 20 micron resolution. So those fluctuations that you see are neural activity that's fluctuating. And a big advantage of this approach is that we can measure neural activity through an intact skull. So we don't have to do any surgery to put windows or anything like that. We can just image right through the skull. And this means it's a rather more high throughput method than methods that we normally use. And it also means that we can study all through the course of learning, which turned out to be very helpful, as you'll see later on. Okay, so number one, we figured out a way to measure neural activity across the cortex. Next, we had to figure out if animals can make decisions in this new setup. Well, why is that a big deal? I already told you, we're studying decisions all the time, human, rat together. The problem is that in the microscope that I showed you, we need to have animals that are immobilized and not moving because the microscope can't really follow them around when they're making movements inside the big behavior box, like in the movie that I showed you before. So we had to adapt our behavior and training procedures away from a freely moving rat into head-fixed mice. And we had to switch from rats to mice because we needed to have the genetic tools that are available with mice. Remember, the point of this approach is that we're measuring these calcium signals across the cortex. Well, that means we need to have the calcium indicators all the way across the cortex, and pretty much the only way to do that is with the genetic strategy. So this was a big change, but we made a big shift. We retooled the lab and altered our decision-making behavior so that they were suitable for animals that were in a head-fixed state. The kinds of decisions I'll be telling you about today are pretty simple ones relative to the other ones that have shown you before. They are spatial localization detection tasks where the animal is presented with either an auditory or a visual stimulus on the left or on the right, and the animal's job is to make a licking movement to the side corresponding to where the stimulus was. This is a somewhat simpler task than the ones that I've been telling you about so far, but we've recently extended this to a discrimination version where there are stimuli on both sides and studied a lot of these questions in a cell type specific context. So if you're interested in that, you can look up that paper on the bioRxiv. We placed it there pretty recently. Okay, so I'm going to show you some new movies now. This is, again, animals making the decisions, but this time in a totally different context. So you're seeing now an image of a mouse, and the camera is underneath the mouse, so you're kind of looking from the bottom up. That's its little nose and its whiskers at the top. And at the bottom of the image, you can see two handles. And when the animal initiates the task, the two handles move in, and the animal has to make contact to tell us that it's ready to make a decision. Now you're seeing the animal from the opposite point of view, this time from the back. That's its little tail hanging down at the bottom. And I'm going to show you now the stimulus period. There are going to be two visual stimuli that will appear in succession on the right. Okay, I think the video got cut short. That might be only one, but anyway, hopefully you all saw it. Okay, after that, the animal has to hold its decision in mind for a second. So we have a little bit of a working memory component there, and then it's time for the response. So what you'll see in this movie is the two lick spouts at the top of the screen will move in, and the animal will lick one of them, and the other one will move out of the way to prevent the animal from changing its mind. So that's how it reports its choice. Okay, so this was encouraging. We were able to train up a head-fixed decision-making task in mice, which was pretty new to us. We were encouraged that was possible to do. So now we were finally ready to measure neural activity and behavior together in the new setup. So I'm going to show you now a couple of movies that are pretty raw data, so you can see what the data looked like as it was coming in. And you'll perhaps in your mind start to understand some of the questions that we asked when we were looking at this data as it was coming in. So this is the same kind of movie that I showed you before, where the color on the brain indicates the amount of neural activity. But on the left now, there's a video that was recorded at the same time as we measured the neural activity. So you can see the animal's behavior and also the neural activity at the same time. So one of the things that was surprising to us, and there were two total, but the first thing that was surprising is that the activity is really quite widespread. So remember, these are pretty simple decisions. The animal's just like getting a stimulus on the left, licking to the right, but the whole brain seems to be active. There are many, many structures recruited, including the olfactory bulb, which is at the top of the brain. So it was kind of surprising to see so much activation there. I'll keep playing the movie, and you can see that the activity kind of ebbs and flows, but is overall pretty widespread, especially around the time that the animal makes its decision. So this was the first thing that surprised us. Why was this activity so widespread, even though the animals were making pretty simple decisions? The next thing that surprised us will become evident when you look at the activity from 4 trials played at the same time. So these are 4 decisions, and they're different decisions. They might be auditory or visual. They might be left or right. But in most cases, or in each case, I should say, the animal's doing pretty much the same thing, right? They're in the setup. They get a stimulus. They lick to one of the spouts. They get some water. But if you look at the neural activity in these 4 trials, you'll see that each one is pretty different from each other one. So these were the 2 things that really stood out to us when we looked at this raw data, that the activity was very widespread and also that it was highly variable trial to trial. So returning to the checklist, we were able to measure neural activity and behavior together in the same setup, but we really needed to see whether or not it made any sense. So to do this, our goal was as follows. We wanted to be able to do, for every pixel in the image, to look at the activity on trial 1, trial 2, and so on, and then build a model so that on held-out trials, we could predict the activity based on all of the stuff that we knew was happening, all the behavioral covariates at our disposal. And we had a few. So for example, on each trial, the stimulus comes on at a different time. There are handle grabs and licks that occur with different frequencies and at different times on each of these 3 example trials, because those are things that are under the animal's control. And we also measured video data as animals were engaged in this trial. And I've showed you here just the top 3 PCs from the video on each of these 3 trials. Of course, if you look at these, they don't really mean much. They didn't really mean much to us either. But we thought, well, what the heck? Let's just include them in the model. And maybe there'll be something in this video data that's helpful in allowing us to make sense of this neural activity. So we took all of these behavioral regressors together and put them in a big design matrix and used L2 regression to try to find the weights that best mapped all of the stimuli to the neural activity. I was going to do a slight digression for aficionados. I think I won't, but if you're worried about the fact that some of these regressors are discrete and others are analog, please ask me during the discussion and we can talk about that. So back to the model, I'm going to show you now the prediction of the model on these 3 example trials. So these were held out trials. So we did not fit the model to these trials. We fit the model to other trials, and then we test how well the model can predict activity on trials that it has never seen before. So I'm going to overlay that now in red. So look to the right side of the screen. And you can see that we were actually able to capture a lot of these single trial fluctuations pretty well. I was quite surprised when I first saw this because, after all, cortical activity is notoriously variable trial to trial. And in fact, a lot of that variability, well, people have many theories about it, but one possibility is that it reflects an entirely stochastic process. But if that's the case, we shouldn't have been able to model so many or predict so many of these single trial fluctuations. So I don't expect this crowd to be convinced by 3 example trials. So I've showed you now down here how well we can predict the activity on single trials. These are all held out trials across the brain, averaged over all time points. And overall, we were able to explain about 42% of the single trial variance, which is a lot more than we expected. And it also put us in this kind of cool position of being able to say, okay, now let's look under the hood of the model and let's see what's making the model work. So in other words, which behavioral variables drove the model's success? So the way that we look at this is to take all of the variables that we included in the model, and there are many, and then to compute for each of this what we call the cross-validated R-squared or CVR-squared. That's really simple. It's just the correlation between what we predicted that the neural activity would do and what it actually did. And the CV is just for cross-validated. So we can get a value for each of these task variables here. And this group of variables are all the things that any self-respecting decision scientist would include in an experiment. Things like when the sensory stimulus came on, whether or not it was a rewarded trial, previous success, current success, and so on. And these variables, accounted for a certain amount of single trial variance, so that was fine. When we looked at instructed movements, the things that we taught the animal to do, like grabbing for the handles and making the licking movement to the spout, we found that those accounted for quite a bit of variance. But the big surprise was this. So when we looked at the amount of variance accounted for by uninstructed movements, things we never even taught the animal to do, and in fact didn't even really know the animal was doing, we found that they accounted for far more of the variance than any other variable that we looked at. They really dwarfed the task variables, which were the things that we were really interested in studying. We were naturally very skeptical of this result and sort of tried to do everything that we could to break it. And I can tell you about a lot of the controls and other experiments we did in the discussion if people are interested. Just to tell you about one of these, we realized that we needed to do a much more conservative analysis because, of course, if you're looking at these variables on the slide at all, one thing that might jump out to you right away is that many of them are related to each other, right? And we tried as best we could to orthogonalize these to each other, but it's kind of impossible to do that perfectly. So in order to figure out the unique contribution of each regressor, what we would do is take that regressor, throw it out of the model, refit the model without that regressor, so basically repeat the whole process, do all the cross-validation and everything, and then see how much worse does the model do. You might call this like kind of a model suffering index. And if the model does much worse, that means that your variable was important. So when we did this more rigorous test, we found that many of the variables did not really survive it. So that means that if we kicked them out of the model accounted for almost as much variance as it did if we included the variable in the model, which was kind of surprising. But the variables that really did survive were all the things that were uninstructed movements, especially the video and the video motion energy, which is just the first derivative of the video. It just sort of tells you how much overall movement there is. So to summarize, we took all of these variable groups together and pooled across the variables. And you can see here that both with our more rigorous and less rigorous tests, that uninstructed movements always dominate neural activity. This was true for every moment in time in the trial. You can see that the black line is always above the blue line and the green line, indicating that uninstructed movements always dominate. There are little dips at the time of instructed movements, where those instructed movements very briefly account for some of the single trial variance, but even then they don't even really come close. And here's a movie showing you unfolded over both space and time the contribution of each of these variable groups. So in a moment, the visual stimulus will come on. The task variable briefly explains activity in V1. But overall, instructed movements appear to be the most important. Okay, so that was the last item on our checklist, to analyze the neural activity to see if it makes sense. And it sort of did, except that what we expected to find was something really different. We really had not anticipated that movements would account for so much neural activity, and we were really very surprised to see this. So our conclusion from these observations is that movements dominate neural activity, even in trained experts. And in other controls that I won't have time to tell you about, we dug deep into this result, first replicating it with 12,000 single neurons that we measured using 2-photon imaging, and then also with electrophysiological data that we collected with neuropixels probes. And every way we looked, we found that the movements always dominate neural activity. And this kind of represented a change in thinking of us, both in terms of interpreting brain activity, because if you know that most of single trial variance is driven by movements, that affects greatly how you interpret that trial-to-trial variability. And also, I think more broadly, thinking about how decisions and movements or cognition and movements might be connected. I think many of us tend to think about being engaged in a cognitive process as requiring one to be still, right? After all, the thinking man is himself made of stone and isn't moving at all. And we tend to think of cognitive processes as being rather separate from movement processes. But in fact, in real life, maybe that's not so true. And in fact, humans tend to fidget a lot. And there's been some recent papers suggesting that human fidgeting data corrupts neural measurements in humans as well. And also, if you just look around the room or look at your fellow passengers on the subway, people are moving all the time. So it's made us wonder whether we should think more broadly about how movements and cognition are connected. But I think sort of stepping back, when we started to look back at what we had discovered and wonder why this hadn't been reported previously, I think there's a few reasons. And one of them is that we used many more movements than previous studies had included. And we also used them in kind of a hypothesis-free way, right? So rather than saying like, well, I think the nose is important for decision making, so I'm going to track the movement of the nose, we said, well, we don't know what's important for explaining neural activity. We're just going to take all the video data, squish it down a bit, and just put it in the model and just see what the model happens to find. So that was kind of the first difference. The second difference. The first one was the number of movements. The second one was the hypothesis-free. And I think the upside of our approach is that we learned something that we really didn't expect, which is that movements have a much bigger role than we had anticipated. But the downside of that approach, that hypothesis-free approach, is that we're kind of left not knowing what really happened, right? We just put the video in the model, accounted for a lot of variance. What was the animal actually doing that mattered so much? Well, getting to the bottom of that has been a thorny issue, and is something that we're still really working on in my lab. As a first pass, one way that you can relate the neural activity to the video data is by looking at the weights that were assigned by the model. And so we can project those back up into the full dimensional space, because of course in the model we don't use the whole video, we use the top 200 PCs. And then the pixels are colored based on how important they are for explaining neural variance. And I don't know, it's like sort of useful, right? There's clearly some facial movements that are occurring around the jaw and the nose. One possibility is that the animals are making facial gestures that mice are indeed known to make, and that those are related to something it's experiencing in the task. We don't know. We're working hard to get to the bottom of that, and also to understand the relationship between the nature and frequency of these movements and also the animal's performance. One approach that we're using has been a collaboration with Liam Paninsky and Scott Linderman and some of their colleagues using a tool called BehaveNet. And BehaveNet is kind of neat. So it's kind of in between our totally hypothesis-free approach and a very, very hypothesis-driven approach where you like track a single part of the body like the nose. Instead, these tools that they're developing try to find behavioral syllables and can track their occurrence and also the transition between them. This is kind of a challenging tool to use in a head-fixed context because, of course, a lot of the syllables that the model discovers are syllables like things we trained the animal to do, so we sort of already knew they were there. But we're working really closely with this crew who are also funded as part of the Simons collaboration on the global brain. And we think that being able to characterize these movements in more detail will help us to understand what they're why they're there and why they are perhaps sometimes hurting and sometimes helping cognition. So as a first step towards closing that gap, we did realize that there was something that we could gain by looking at these movements over the course of learning. So the animals start off as novices on the task and slowly progress to being experts on the task. And we're able to track their neural activity throughout that entire period and also able to track their behavior. And one really interesting thing we noticed is that there were big changes in the behaviors themselves. So first, I should say that in terms of the contributions to explaining neural activity, uninstructed movements always dominate from the beginning of learning to later in learning, but their overall contribution does decrease slightly. And we first considered the hypothesis that the impact of an individual movement changes over learning, right? Like maybe there's a movement that early in learning is controlled by the cortex, but as the animal becomes an expert, it's controlled by some other, perhaps deeper part of the brain that we don't have access to. But we found that when we looked at the impact of an individual movement, that is the beta weights that are found by the model, it didn't really change that much. So it seems like if you're an expert making a hind limb flexion versus if you're a novice making a hind limb flexion, you get pretty much the same response in the brain. So we wondered then, well, what is changing? And so we thought instead maybe it was something to do with the movements themselves. So I have an example movement that I'll show you here so you can see the dramatic change that happens over learning. So on the vertical axis is just the events per trial. It's just the frequency of whisking that's occurring. And on the horizontal axis is time. So the trial is about 4.5 seconds. So that's just the time in the trial. And this is not an interesting plot at all. All this is telling you is that the mouse is equally likely to whisk at every moment in the trial. Not very interesting. However, if you look at expert animals, it's a totally different picture. So expert animals have a very structured whisk. So at 2 seconds, the visual stimulus comes on. They have a very stereotyped behavior. And then they have a second, slightly less stereotyped whisk around the time that the reward is delivered. Importantly, whisking has nothing to do with this task as far as we're concerned, right? We didn't teach the animal to make a whisk. It doesn't harvest rewards or indicate choices by making whisks. This is a behavior that the animal elected to do of its own volition for reasons that remain unknown. So what does this mean for interpreting neural activity in experts and novices? And what does this mean in terms of like actual real movements? Well, so one analysis that we did was to look at the change between task-aligned and task-independent movements. So task-aligned movements are like the whisks, like the whisk that you see at 2 seconds there. They're movements that occur at relative to task events. Task-independent movements are ones that occur idiosyncratically and are not tied to specific events in the trial. And what we found is that over learning, so now the horizontal axis is now training days, so that's the many, many days over which the animal goes from novice to expert status. Over training days, the task-aligned movements go way up. That means many of the movements are now occurring at the same time on every trial. And the task-independent component goes down. That means that those the idiosyncratic, fidgety type movements are decreasing. Okay, but what does that really mean in terms of things that humans might actually do, right? Eventually, we want to tie this back up to human decision-making. What are task-aligned and task-independent movements? How do we really think about them? Well, so I think in human, we actually see task-aligned uninstructed movements quite a lot. And these tend to occur frequently in trained experts, especially athletes and musicians who are very skilled at performing a particular movement and often have many, many uninstructed movements that either precede or accompany that movement. So one example is baseball players who often, as they go up to bat, will make a series of very stereotyped fidgets that to us as observers are not obviously relevant to the job of hitting the ball. See it again. Here's one more. This guy has a really nice fidget. He has to hit both feet twice with the ball, and he does that every time. And from our point of view, this seems kind of odd. Why would athletes and musicians make these movements? They seem to us to not obviously have anything to do with the movement at all. But from the point of view of the person carrying out those movements, they often feel like they're very much part of the learned skill. And I don't know why that is, but I think there's a pretty natural explanation, which is that with movements that are hard to learn, you tend to gradually become expert at them through the mechanisms of reinforcement learning. And so that means when you happen to get it right, early in life in the Little League, you happen to hit the ball, your brain says, okay, that was good. Whatever we did, we got to do that again. And your brain doesn't know exactly what it was that led you to hit the ball. So you naturally have an imperfect estimate, and you end up having movements reinforced that aren't actually related to the movement at all. And this is probably something that happens in musicians as well. And these sorts of task-aligned, uninstructed movements have been reported previously in animals, kind of in a different context. Often they're used to kind of time delays, right, as if you're sort of tapping your foot to time a particular delay, but they're probably of the same sort. The movements that I think are perhaps even a little more interesting are the task-independent, uninstructed movements. And humans make these a lot. Leg rocking, leg shaking, chair rocking, hair twisting, eyeglass, I don't know what John Krakauer's doing, various other things. Humans make a lot of task-independent, uninstructed movements. These are energetically demanding. They also often come at a social cost. People tend not to like it when other folks are fidgeting. And yet, despite the energetic demands and the social costs, they're very, very hard to suppress. So I don't know why that is, but it makes me wonder whether either these movements are benefiting cognition in some way, or they're reflecting the fact that the subject is aware that their cognition or their focus is slipping, and they're using these movements as perhaps some kind of way to kick the cognitive machinery into gear. And it's just a hypothesis, of course, but I think my overall rationale for thinking that is that ultimately our brains evolved to move us around. So it's kind of a natural idea that our cognition might be kind of taking advantage of mechanisms that were already there to be used for movements and using them in a different way. We don't know all the details about how that might work, but it does suggest to me at least that cognition and movement might be much more closely intertwined than was previously appreciated. Okay, so I've told you so far that studying human and animal decisions together reveals common computations that are shared by diverse brains, that rats, like humans, can flexibly change how incoming signals are weighted to combine information optimally, and that even when subjects are experts in these decision-making strategy, their overall activity is dominated by movements. And more recently, we've replicated that finding in a cell-type specific context where we measure the activity of single specific classes of excitatory pyramidal neurons, and have found that if anything, in certain classes of neurons, the tendency for movements to dominate is even more apparent.